Okay, I think we are ready to go. Okay, I'd like to begin by acknowledging and paying my respects to the traditional custodians of the land on which each of us are today. I would like to pay my respects to their elders, past, present, and emerging. Let us pause to learn, acknowledge, and celebrate their ancient histories and cultures. Welcome to the third seminar in the Deacon's Science and Society Healthy Futures series. My name is Neera Bhatia. I'm an associate professor in the School of Law at Deakin University and co-theme leader of the Healthy Futures research theme. The Science and Society Network was formed in 2018 in recognition that scientists and humanities and social science researchers need to work together to meet the great challenges of this century. No single academic field can bring about the changes we need to see in the world. Bridging disciplinary divides is key to finding new solutions to the problems we face. The Science and Society Network supports early and mid-career researchers across the university who are embarking on groundbreaking interdisciplinary projects. For those of you that are attending our seminars for the first time, here's a quick rundown of the structure of today's event. I will introduce our speaker, Dr. Charles Dupras, who will be presenting on the rise of consumer multi-omics, emerging concerns. Charles will present for 30 minutes or so, and then we will have discussant comments from Dr. Chris Mays from Deakin University and Associate Professor Jeff Craig, also from Deakin University. I will formally introduce both Chris and Jeff a little later. We will then open for general question and answer from the audience and general discussion. I'll be chairing this discussion here on Zoom. My colleague Tao will be moderating the YouTube live chat. We're keen to hear from you and answer any questions you may have. So you can either post those on the YouTube chat, or you can send your questions to us on Twitter at SSN Deakin, and make sure you use the hashtag SSN Seminar or Healthy Futures. Now to our speaker. Dr. Charles Dupras holds a master's degree in molecular biology and a Ph.D. in bioethics. He's currently an academic associate at the Centre of Genomics and Policy at McGill University in Canada. Charles's work touches on potential non-medical applications of epigenetic technologies in different contexts, such as insurance, immigration, forensics, direct-to-consumer testing, as well as national defence and security. Over to you, Charles.
Thank you very much, Neera. Okay, so I'm going to share my screen here. Here we're going to go. Okay, thank you for the kind introduction. I would like to thank very much the Science and Society Network and the team at Deakin University for offering me the opportunity to present this work today. So the rise of consumer multi-omic testing. Most of you may be familiar with direct-to-consumer genetic tests and testing companies such as 23andMe, Ancestry, Gene2Gene, and others. You may be less familiar with new types of testing that are being offered online to consumers, and these are epigenetic tests and microbiomic tests. So we're going to touch a little bit to the practices of these companies today, and also we're going to discuss a little bit the ethical, legal, and social issues that come with the rise of these companies in recent years. Most of my talk today is going to be about a project that we conducted for about a year, and it was funded by the Office of the Privacy Commissioner of Canada. So we proposed to do content analysis of the websites, basically, of these companies, these new companies, and of their terms and conditions, their privacy policies, and their promotional messages. So as you can see here, we've already published a paper that's titled "Selling Direct-to-Consumer Epigenetic Tests, Are We Ready?" In Nature Reviews Genetics in January. So that was preliminary results. We've been working on more of the qualitative part of this research, and it's soon going to be available for publication in another journal. And so we're going to talk a little bit about these results today. So that's the empirical work that we've done. On a more conceptual side, what I'd like to propose is to start thinking not only about, we've been talking about post-genomic times for almost 20 years now. I want us to start thinking about the multi-omic era, okay? There has been, over the past few years, we know an accumulation of omic data in large-scale databases, which we can call the multiplication of data. There's been international circulation of omics data, which we could call multi-jurisdiction data. Private and public research databases are now sharing data, so it's multi-sectoral data now, really. But there has also been an unprecedented diversification of omics data in these databases. So that's what we call data multiplicity. And as we mentioned in another paper that's we've been working on lately, with my colleague Eline Bunic, we said, while the accumulation and increased circulation of genomic data have captured much attention over the past decade, privacy risks raised by the diversification and integration of omics have been largely overlooked. So what is multi-omics? Okay, so we conceive it as attention not only to genomics, but also to, of course, epigenomics, transcriptomics, proteomics, metabolomics, and microbiomics. So all these complementary research fields, which also are interested to biological variants in the human body that can tell things about risks to diseases, but about how your body works, basically. And there's an increasing number of research who look at actually the interaction of these different systems, and so they perform what they call integrative analysis, and they can do this in single cell analysis, for instance. And the merging and the integration of these different biological systems is quite complex to study, actually. And they scientists have started to implement machine learning technologies to help them doing this. One of the great examples of epigenetic variants and research on epigenetic variants is epigenetic clocks, okay? So this is research that has been conducted on biological markers on the DNA. It's not really in the sequence of DNA that we look now, it's over the DNA. That's what we call epigenetic variants. And they look at variations in terms of the methylation of DNA, and when combining methylation levels at different places on the genome, they can predict pretty well your epigenetic age, okay, your chronological age. But there's also a way to measure variation between your differences or discrepancies between your chronological age and your actual biological age. So there's a possibility that, let's say, you may be aging faster than what your age actually shows on your biological passport. This has been related to the possibility of predicting your life expectancy, because if you're aging faster, then the possibility that you will live longer is shortened in a way, okay? And this acceleration has been linked to diseases. It has been linked to acceleration factors, such as exposures to pollutants, stress factors, and other chemical products that you may find. There's also prospects of technologies that may be able to rejuvenate epigenetic age using pharmaceutical products, but that's a little bit more futuristic, let's say. When we talk about microbiomes, we talk a lot about gut microbiome. So that's a way using genetic technologies to detect the microorganisms that are in your gut. So bacteria, viruses, protozoa, fungi, you can detect them, not only their presence or absence, but also the proportions in which they populate your gut. Using metagenomic analysis. And these different proportions have, or the presence of some microorganisms or absence have been associated with some diseases. And these have also been associated with exposures and lifestyle, diet, for instance. Here you can see, I'm showing a little table of this is in November 2018, when we first proposed to do a project with the Office of the Privacy Commissioner. There were, we have identified not more than 3 companies were actually offering these tests. And as you can see, when we actually started the project, there were already 12. So we did our work on 12 companies. And later you can see in March 2020, earlier this year, there were already like almost, I think, 8 more companies that had seen, that had stormed into the market. You can also see that Chronomix, one of them, had started to offer tests for COVID, and also that Biome had diversified their offer by offering not only microbiomic testing, but also epigenetic testing. So you can see we can start talking here about multi-omic testing or multi-omic databases owned by the same company. So here you can see that's just an example of a company, MyDNH, which offers you, well, they say they can reveal your biological age through epigenetics. They say they own the most accurate biological age test that is based on Dr. Horvath's epigenetic age clock. And that's pretty, I mean, that's pretty descriptive. They don't talk that much here, at least they don't assume that they're going to tell you, they don't say that they're going to tell you health information about yourself. But here it is a little bit different. Chronomix says literally that it's going to free yourself from health worries, and that you can track and reduce your risk of future illnesses with their award-winning epigenetic testing solutions. So that's another approach here. And here is another page on the website of Chronomix, where you can see that there are different kits that you can buy, let's say. You can either go with the cheap kit, which is somewhere about $500, where you will have access to a yearly epigenetic test, and you will also know about your biological age. But you can also become, as they say, a genome rock star by also including a genomic genetic test in your kit, and that's going to reveal much more about you. It's going to reveal not only is it going to provide you with your whole genome test and information about your full genome, as they say, but also about health indicators. So they talk about biological age, they talk about the metabolic state, and they talk also about smoke exposure, okay? So these are what they offer. There is also another step after testing, after monitoring. Some of these companies, they will propose you to, or they will recommend some here cosmetic products that better fit your epigenetic profile. So you will see, they say in this context that they match cosmetic products with your skin's aging. Whether that works, is unclear. Okay, we can discuss that later. Here, similar approach, but with microbiomic tests. So not epigenetic test, microbiomic test. They'll offer you, it's a little cheaper here. They will send you a kit. You will be able to, in the context I forgot to mention, but most of the time it's going to be saliva sample that you'll send for epigenetic tests. Here, it can be stool sample, for instance, and they will be able to detect the composition of the microorganisms that populate your gut, and they suggest that you will be offered or that you will be empowered, that you will be able to eat better, knowing how what microorganisms compose your gut. Here they say treat, it's strive here. They say treat root causes, not symptoms. Here a concern could be that, is there a risk, for instance, that people who see that will say, okay, well, if I use these probiotics that this company recommends, I shouldn't be concerned too much about the symptoms. Okay, I'm having a I'm actually choosing to use natural products instead of validated standard of care that could be recommended by my physician, so that you can start seeing here that there might be some ethical concerns that arise when we say treat root causes and not symptoms. But we're going to talk more about this a little later. So we see a sort of a shift, or at least an expansion in the industry here, from tests that were previously done on your inherited biology, right, with genetic tests, to tests that are being done on biological markers that can change, that can be reversed, probably if you change your lifestyle. It sort of provides a new picture or a new window on people's lives. So not on life itself only, but on your everyday exposures and everyday lifestyle and life choices. As mentioned by Chronomix, genetic tests miss the impact of lifestyle and environmental factors, which we know can have a profound impact on our health. And Mudo here suggests that, or asks you, would you like to reverse the aging process with the epigenetic tests? And here it is MyDNH who suggests that you can track again your progress with the most accurate and reliable DNAH test. So again, it's seen or is promoted a lot as something that will empower you and that you can act upon, and that you can orient your life according to your biological profile, okay? And that you may also not only act according to your biological profile, but at the same time, you may be able to change it. There are a few ethical and legal issues. I've mentioned rapidly before. Medical relevance is unclear. It is often products that are being sold as being medically relevant, but there seems to be a lot of overstatements or exaggerations in terms of their potential benefits for the health and welfare of people, well-being of people. But most concerning to us is the inconsistencies that we can see in between the promotional messages of these companies and what is really included in their terms and conditions and in the privacy policies. Now, let me give you just a few examples of these statements we find a little bit overstated. exaggerations, I should say. So XLS say that they will provide you with clear results and actionable recommendations, and that Thrive says that it is clinically proven health benefits. Well, it's not all that clear that it will lead to a better health outcome if you use these technology. Something that is to us clearly an overstatement here is what they propose when they say that they propose or they suggest that you should perform or use these tests 1 or 2 times a year to track your progress. So meaning that, and this implies actually that their test is accurate or precise enough that the margin of error is so small that you can test at different points in times, and that the difference between the two that you're going to see is accurate. This is really unlikely given the current state of these sciences. And, I should possibly mention that we can understand, or that we can, that we can understand the financial incentives that are behind that kind of recommendations, and that it's part of this new model of tests, that this new business model in which you will not only have now to test once for your genetic profile, but now you can test more than once. So these tests you can use and reuse, and as part of a business model, it is much more profitable. The sensitivity of the information is, as we said, we mentioned briefly earlier, it is somehow biology intrusive, but not only biology intrusive, it is also to some extent life intrusive as well. And it is information that, according to the policy, the privacy policies of these companies, it is not clear for almost half of them whether they consider the biological data that they obtain with their tests, whether they treat it as personal data. That's going to be important because many ethical guidelines and regulations will not apply to de-identified data that is not considered as personal data. Data protection. So there has been some studies that have shown that if you, well, we know for some time that genetic information is so unique that it can be used to re-identify a person. Let's say if you have a database that contains genetic data with your name and another that contains only your genetic data without your name, you can link the two, and using the one without your name, you're going to be able to link back to your name. Okay, so that's a possibility to match because it's so unique. So there's been studies that have shown that whole epigenome sequencing, or having access to many information about many markers on your epigenome may be something that could be used to identify you or link you, for instance, to a database that's a clinical database. So you're going to be able to deduce that, let's say, you're part of a cohort in clinical research, for instance, and for this reason, you may be, some people may be able to tell that whether you have a disease or not, let's say. And there's also a possibility that if you're using a multi-omic database, and that you believe that you've adopted a very strict data protection strategy by masking SNPs or genetic variants on the genome, there has been correlation that have been shown between some epigenetic markers and genomic markers. So using the epigenetic information, you may be able to sort of infer genetic information about someone. So we need to rethink a little bit our data protection strategy when we use multi-omic databases. Also, it's worth noting that the level of sensitivity will will depend, of course, on the information that's provided. And what we know now, epigenetic data can provide as information may change in the future. And that's something that's recognized by the companies. They say this data could have greater meaning in the future as new discoveries are made. And also, Ixela says, non-personally identifiable or aggregate information may be used by us for any purposes permitted by law. As I've mentioned earlier, it is unclear whether epigenetic data or microbiomic data is considered as personally identifiable or not. So we're not sure if whether this applies here. And Thrive says, we may disclose any information about you to the government or law enforcement officials or private parties as we, in our sole discretion, believe necessary or appropriate, including to stop any activity that we consider illegal, unethical, or legally actionable. So that's something we're considering as well, especially if we know that these concerns may be compounded by the fact that the privacy policies of these companies are very inaccessible. They are written in a way, in the way that we write in scholarly literature, basically, and they take almost an hour to read. So these are measures that we've used here to assess the accessibility of these companies. So, and there's also concerns about non-discrimination laws that actually exist for the context of genetic information, but that may not apply to epigenetic or microbiomic information. Because epigenetic information is non-genetic in a way, and because microbiomic information, yes, it's often genomic information, but it is non-human genomic information. So this is just an illustration of the non-discrimination policies that exist worldwide. There's a big variety, and most of them have been adopted because we're concerned about the use by third parties, such as life insurance companies. But as you can see here, companies that have started also to propose epigenetic tests, life expectancy tests based on epigenetic markers that they could use to offer you different premiums. So there are concerns for secondary uses in the context of insurance, but there are also in the context of forensics. There are also in the context of immigration, where public agencies, not only private companies, may use epigenetic markers, for instance, in the context where they would like to prove whether a minor is under 18 or not. It's been seen in the case of the Eldeshin case in Germany. And most recently, DARPA in the US funded projects to develop a tool that's going to be able to assess exposures to chemicals at war. And we can see that national defense and security, the national and defense security context could see an interest in epigenetic tests. So here this is preliminary results from a survey we've conducted with epigenetic researchers. We asked them basically, how concerned are you about these different uses? And you can see here, maybe on the right-hand side, it's easier to see that there were up to 75% of them that were either very or extremely concerned about insurance, 50% for immigration, 50% for direct-to-consumer tests, and 25%, a little less, for forensic uses. So what's concerning is not only that laws are not really didn't give a lot of attention to these new tests, but also that human rights declarations have been adopted and that they've been really designed for the specific context of genetics. And when you look at them, you can show that they really do not provide guidance for epigenetic information and the governance of such data. So you can see concerns at different levels. Yes, direct-to-consumer tests, but also biobanks. How, what criteria should we adopt in this different context for the return of results to participants or of incidental findings in the context of epigenetics? There are questions that we can discuss later about that. Potential misuse, but also multi-omic privacy risks. And this is something that we've started to work on. We want to sort of, again, propose a framework that's going to be focused on the multi-omic landscape that is developing. And so based on the properties that are shared by these different omic types, we want to develop an approach that's going to be sensitive to the re-identification risks and level of sensitivity that appear when we merge data sets of different origins, and also of the interrelation effects between these different data sets. Okay, so there might be correlative or synergetic effects between the data, and that these may also raise privacy risks. So these are just a few of the current research projects. Most of them I've spoken about already. We would love to see a part 2 to our exploration of the website of the companies. For instance, we would love to develop projects that would look at what happens once you've purchased the tests. That's something we haven't done yet. We've only looked at the website and the policy documents. And we could also survey people who actually use these tests already to see what has been their experience using these tests. So most of our, I would say that the take-home message here is that we really need to move away from what has been called by legal scholars genetic exceptionalism. And the father of the term, Thomas Murray, said again recently, yes, genetic information has certain properties that warrant close attention, but nothing that really launches it into some unique universe of moral, legal, and policy concerns. And also Ellen Wright Clayton mentioned, although most commentators have been critical of genetic exceptionalism, virtually all of the recent legislation has been genetic specific. And here, we suggest that this might be a good place to start for companies to sort of improve their practices and their policy documents. This is a document that has been adopted in 2018. It's called the Privacy Best Practices for Consumer Genetic Testing Services. It was a product of the Future of Privacy Forum, and it calls for a certain types of a few conditions that companies should meet, and it's been adopted by a few direct-to-consumer genetic testing companies regarding, privacy policies, specific consent requirements, and requirements for data sharing as well. We should note, however, that de-identified information is excluded from these standards, meaning that we still need to think about re-identification risk when we merge data. Even though they're identified, they might be re-identified, and that's a concern that we should keep in mind too. So I would like to conclude by acknowledging the work of my collaborators. And my team from McGill, of course, Jan Joly, Guillaume Bourque, Ken Dewar, Sarah Kimmins, were great collaborators on this project. And I would like to highlight the great work of my research assistant, with a special mention to Teres Naupers, who has been working very hard on this project, and also, of course, to the funding and to the team here again at Deakin. Thank you very much.
Thank you, Charles. That was an extremely insightful presentation, and I know that we have a number of questions for you. We'll begin with questions from discussants, Dr. Chris Mays and Associate Professor Jeff Craig. Dr. Chris Mays is a DECRA Research Fellow in the Alfred Deakin Institute at Deakin University. His research interests include the history and philosophy of healthcare, bioethics and political theory. His current research is on the history of bioethics in Australia and its contributions to regulatory frameworks, legal reform and public discourse in matters of life and death. He is the author of Unsettling Food Politics, Agriculture, Dispossession and Sovereignty in Australia, and also the author of The Biopolitics of Lifestyle, Focal Ethics and Healthy Choices. We'll then hear from Associate Professor Jeff Craig, who is a lecturer in medical sciences at the School of Medicine at Deakin University. Prior to working at Deakin University, Associate Professor Jeff Craig spent 20 years as a researcher at the Murdoch Children's Research Institute in Melbourne. He studies the role of epigenetics in mediating the effects of early life environment on the risk for chronic disease, and he is currently developing epigenetic biomarkers for easy to collect biosamples. I'm sure there are lots of questions from both of our discussants, so I'll hand over to our discussants to begin with, and then we will take questions from the audience, which I am already receiving.
Excellent, thanks a lot, Nera. Yeah, and thank you, Charles. I mean, that was a really fascinating series of studies that you've been doing in this area and on epigenetics and their consumer application. There are a number of different responses and questions that I have, and so it'll be good, yeah, to have a discussion around some of these things. And particularly, I guess it brings me back to some of my research on the biopolitics of lifestyle and the way conceptions and understandings of lifestyle in relation to disease and health have been, I think, amplified in this post-genomic context, or providing an epigenetic overlay to some of those interventions. But before getting to that, I guess my first thought is how we are in the market so clearly and presently with the application of these research in epigenetics. Obviously looking at direct-to-consumer testing around these omic technologies. And that's surprising. I mean, it's very unsurprising in many respects, just the dominance of the market and the need to translate research. But also, and we can get to definitions of epigenetics, but one of, I guess, my concerns or frustrations is that this field of study and field of science is wanting to move away, or move towards, I should say, the focus on the environment and history in the shaping of health and health outcomes. Yet so much of this is focused at individuals, individual interventions, individual choices. And I guess further to that, in the context of the market, and you did refer to some surveys of researchers, but you know, the research agendas in the university, both of STEM and humanities scholars, are maybe not explicitly driven by the market, but there's certainly market signals put in place by government funding bodies, and the desire for translation and the need to show how this research can directly be translated into market or consumer areas. So I guess my first point would be to bring this together. On the one hand, the direct-to-consumer focus on individuals, and also the research agendas perhaps driving some of these applications, is there seems to be no social determinants of health application. So similar to the stuff on lifestyle and looking at, say, chronic diseases and nutrient deficiency, well, sorry, not nutrient deficiency diseases, but diet-related chronic diseases, there becomes this, instead of having a focus on broader environmental and social factors, there's this focus on individual factors. So perhaps facetiously, it'd be interesting to know if any direct-to-consumer tests also provide a kit to go out and test your soil to see if there is heavy metal content in the veggie patches that you're growing your food in, and also, you know, looking at food deserts and these sorts of things as impacts and other environmental factors that you and Jeff are no doubt aware of. And this comes to then, I guess, the question of a lot of this seems to be diagnostic as opposed to therapeutic, heavily diagnostic. We can show you all of these amazing things about your history and your future. But ultimately, like with former Human Genome Project projections, the therapeutics come back to this very everyday interventions. Change your diet, do more exercise, and buy this highly marked up cosmetic product. And I guess, yeah, it'd be interesting to see outside of this consumer focus, to hear, again, Jeff may have some insights on this as well, whether there are sort of clearer sort of social determinants of health and social justice applications of these kinds of processes. And I guess just finally on that, like a lot of lifestyle interventions in the past, and this lay understanding, I think that this is interesting with epigenetics. I think it does map onto this lay understanding of disease. People are this way because their parents were that way. There does seem to be an intuitive understanding of some of the epigenetic knowledge and information, but it's also sort of applied to these very, I guess, the very vague and ambiguous context in which it's applied allows these companies greater flexibility to present their information without any sort of clear, concrete examples of progress. Like, come back in a year, let's see how your future projection of heart disease is going. Even if it's as concrete as that. I mean, it's similar. There's an article I really like from, I've forgotten the year, but the title is Opening Pandora's Cereal Box. And it was in relation to advertising around cereals starting in the 1980s about how they could, you know, all bran will help your heart disease and these sorts of things. And because we're talking about such lengths of time and such conditions which have so many causal factors to them, you could say anything kind of contributes to a minimization or maximization of a particular disease or health condition. So I think it's great, the work that you're doing to hold some of these companies to account. And it would be good to, yeah, I mean, I'll leave it there, but yeah, I mean, the other things that you flagged around the potential uses in immigration as also frightening.
I would like to, I think it'd be a good time for Charles to address some of these issues where they're fresh, and then we can come back to a point from me and then go to questions. Is that okay with you, Charles?
Yes, thank you. Thank you very much, Christopher, for your interesting discussion of, and you're totally right. I mean, I guess people like to biomonitor themselves. It's not something that's new. You know, when you buy a watch that can track the number of steps you do a day, it's something that's probably similar to tracking your progress, your lifestyle progress in a way. And that's we could present this as something that's new at maybe the molecular level. But yeah, it is different from genetics in a way, because you don't really track your genetic progress. Yeah, there's something interesting about these new tests, which are offering you to know more about yourself at the molecular level, and that I think is fairly new in that respect. So, but I mean, I think you're totally right in saying that there are ongoing trends that have been influencing the way we think about science and the way we think about biology. And there's a paper we published in the Hastings Center report back in 2016. And it's really a paper in which we sort of we're trying to be the Delphi oracles a little bit. So we sort of we discussed, well, a paper that had been published before was sort of making the argument that we should use knowledge about epigenetics to promote all these nice preventive public policies that will help lower health disparities between populations. But we sort of reflect on this, and we were concerned about the fact that we needed evidence at the molecular level to convince us of this. And we started reflecting on this and about the ongoing sociological trends that were in the background, in the landscape of all this these knowledge translation practices that we do, and not only in terms of knowledge communication, but also in terms of implementation. And we had identified two important trends. The one that's the molecularization of health, and also the biomedicalization of life. So Sarah Shostak had worked on molecularization, and Adele Clark has done with a team of sociologists work on this biomedicalization trend. And that sort of had us thinking about really where is going to be epigenetics in a few years from now. And it led us to be a little bit more pessimistic about what it will be. At first, we thought it was great, because when you compare it with genetics, it's great. You see the impact of, yeah, exactly, society on your biology. But at the same time, you had that double, as you pointed out, you had that double possibility, dual possibility. So it's whether you put the emphasis on your biology being determined throughout your life and also throughout the development by external factors that are sort of chosen by society, or you put more focus on the individual. And that is really something that indeed is being promoted by direct-to-consumer companies. They say that it's going to empower you and that you're going to be able to take care of yourself. And so that's really something that we're sort of seeing what we predicted a few years ago sort of materialize at the moment. And I mean, I'm not sure, how we can, if we should, or how we can sort of resist to that trend, because it's difficult to sort of trace back, or make the link between social determinants of health and what has been called, the multiple the determinants of health, and also the developmental origins of health and disease theory, and all these complex factors that have an effect on your life, and sort of go from this really macroscopic sphere to the molecular scale, and navigate between the two, because there's so many confounding factors. I think there's something in the causal relationship between all of these that makes it more difficult to navigate between these different scales, I'd say. But I've said too much, so I'll leave it open for others to comment.
Thanks, Charles, and thanks, Chris. I think, first of all, Charles, I congratulate you for being a pioneer in the area. It's kind of really inspired us in the gene equality group to study these areas, and we'd love to work with you in the future. and provide the Australian perspective as well on some of these tests. And also to Chris, I've been thinking about the issues you've raised recently and been asking around a couple of people within Deakin to get some more ideas and inspiration for discussion. So I'll be definitely knocking on your door or knocking on your Zoom window at some point. What I was going to raise mainly is one issue that's been it's been troubling me, but it's been a focus of my thought over the last year or so. And it was, I noticed that, Charles, in one of your previous papers, you said judges don't like population studies. And there's a big issue for me in when people extrapolate from population studies to personalized studies. To cut to the chase, people like Cecil Janssens and Nigel Panath and a couple of other dissenters have poked holes in the genetics personalized medicine polygenic risk score idea. That there's too much variation throughout most of the risk spectrum of any risk score. And yes, most of these tests will be based on risk scores. Where are you between the least risk and the most risk? There's so much variation for the individual that what these people are saying is that individual data is just about useless. And so unless you're really high risk, for example, and there's examples with breast cancer, for example. And so I'd really like to have to think about how much first of all, not just whether the tests are based on scientific knowledge, as you've mentioned. The first reference I remember looking up in that skin test website was from an in vitro study with throwing things into cell culture, and how that was relevant to humans, I have no idea. But also this, a fundamental discussion of whether the scientific integrity of these tests is valid, and if it's not valid, if they're not actually valid for the individual, then how are we going to pass this information on to the public? I know, for example, that in the field of sports genetics, I know that there was a moratorium passed by probably an international, but at least an Australian group of sports genetics researchers that said the direct-to-consumer sporting ability tests are not good. We don't recommend taking them. Now, in gene equality, we haven't gone that far. What we'd like to do is to inform people, to provide individuals to be able to choose to take these. But I'd like to know your thoughts on how important the technical aspects of these tests are, and how far we should go. Should we go as far as having a moratorium?
I think that there has been studies that have been conducted in the context of genetic tests, and I mean, regulations have appeared after some time of practice. After big companies had even started commercializing these tests, and also there have been studies really with the people who use these tests. And so far, most of these studies say that consumers, they like to be able to have access to information about them without necessarily going to see a physician. And we can understand that people are concerned about privacy risks in the context of with companies, but also when they go and provide biospecimens in the healthcare sector as well. So I think what's important, what has been raised as really important, is making sure that consumers are not being deceived, and this is what they also have. They also agree on that. I mean, they want to purchase these tests themselves, but they want these tests to be accurate and precise. So there's some work to be done really on, I guess, transparency is something very important, because even ourselves, I mean, we know a lot about epigenetics, we know a lot about the biology, but we're not even able to know what they really test for. When we look at the websites, at least, for most of them, we're not even able to know what epigenetic variants or what kits they use. So this is something that I think even companies would gain in being more transparent and showing transparently the level of validity of these tests, because consumers, that's what they want. And if you deceive them, you're gonna lose trust, and it's not good for the whole business model either.
That's a good point actually you make about if maybe a compromise or a joint issue is to aim is to provide consumers and make them and advise them to demand this evidence themselves, as you were alluding to.
Mm-hmm. And I'm sure that scientists could be involved in this. I mean, researchers in the public sector could be involved in validating these tests. You know, there is discussion about different methods, but can we know about the methods we use? If I mean, even consumers are being increasingly aware, and they're more able to search through scientific literature to verify if it's really based on good validated results. And if you just say what kits you used or what markers you look at least you're offering the opportunity to consumers to make informed decisions and better informed decisions. So that's something that I think should be worked on, and really making the privacy policies and the terms and conditions much more simple. I mean, they can have long documents as well, but there should be some page where at least the basic information is simply provided in a simple way, so that everyone can understand well, you know. So that's not always the case.
That's a great segue, Charles. I've got a number of questions from our audience. Can I please ask you, Charles, to bring your slides to stop sharing your screen to begin with?
Yep.
Thank you. Great, fantastic. So I've got about 3 questions. The first question is from Ian Charist. And Ian's question is, do any of these companies ask for your signed consent to use and or to share your personal biological data when you join?
I'm sorry, can you repeat it again?
Sure, I think this is in relation to perhaps direct-to-consumer testing. Do any of these companies ask for your signed consent to use and or to share your personal biological data when you join?
Okay, so to sign a consent form, let's say, okay, we haven't gone as far as actually purchasing the tests, so that's a limitation clearly of our work, and we want to do, as I mentioned earlier, a second phase of the project where we can know more, because at the moment I cannot tell you that they don't, because they may do it later. So that's something to double check.
Okay, great. I hope that answers your question, Ian. A second question has come in from Catherine Metcalf, and Catherine asks, do you think the data accumulated by these companies are likely to enter academic research contexts? Thinking of Rosie's comment that BioValue ties the factory to the lab and 23andMe data mobilities.
It has been part of the business model. It's a practice that has been part of the business model of direct-to-consumer genetic testing companies. So it is likely, yes. I cannot confirm that it is the case. We'll have to look at this more.
And we have another question from Amy Lochman. This question came in via Twitter. And Amy is asking, many of these companies are founded by scientists and are based on some research. What is the level of evidence required for the translation of research findings to direct-to-consumer offerings? And how do we define and quantify this?
That's a big question.
It is a big question.
It is also a very scientific question, and it will depend on you know, the type of evidence that has been accumulated. If you're talking, for instance, about if you want to develop a tool that will assess your biological age, and that you develop this tool or this method with a sample of people or research participants that are mostly of, let's say, white European ancestry, then you can question, or you can say, yeah, it has been validated in this population, but it hasn't been in other populations, and there might be false positives and false negatives depending on who tests. And so there is a need for studies that are on bigger samples, more diversified samples, and also that are being reproduced by different labs too. So it's a big process, improving the precision and accuracy of tests, biological tests, and it is also there's a need for these to be validated, and yeah, it is a long process. But to tell you what's the threshold, I mean, it depends on what's offered to, you know, if you propose a test that is telling you what it's going to be precise, but not very much, then it's fair. It's not deceptive. But if you argue for, or if you suggest to test every week, or well, that's not the case for now, but if you suggest to test every 6 months, then you have to make sure that the test is precise enough to allow for these temporal comparisons. And that is, it depends on what you offer.
I have a question for you, Charles. I have a law background. I'm based in a law school, so I'm interested in what you think are the greatest, I guess, legal barriers or legal implications when we're looking at direct-to-consumer testing. So I can think of some myself in terms of issues around consent and data breaches. From your research and your expertise, what have you come across, and what do you think are the greatest legal challenges or barriers or I guess risks that you think we currently face, and how do you think that they can be, I guess, mitigated? That's probably a very broad question that you can't answer.
No, but I like to, it would give me the opportunity to discuss something that is in my mind, and that I have to confess I am not a bioinformatician, or I'm not even an informatician, but I'm starting to be a little bit concerned about re-identification risks when we merge together that amount of data together. I mean, I'm concerned because most laws or most policies are explicitly saying that they do not apply to data that is not identifying. So, you know, there's different approaches to that question, but whether it is potentially identifying, if it is potentially identifying, should it be considered as personal data and be treated as such? It is a big question for me, but I'm starting to think with the integration of all these multi-omic data set or all these omic data sets together, I mean, there's really a risk that you're going to be able to re-identify someone without even having the names attached. I'm going to give you just an example. There has been some work that has been done using genetic profiles, that you can use the genetic profile of a person and predict facial traits in a way that's, okay, but not there yet. So that means that if you add other layers of information, though, you might get a more precise results. If you add proteomic information, if you add epigenetic information to that, you will, I'm going to give you the example that I have mentioned in the talk. If you have this facial portrait of this person that you've predicted, but you add the layer of the biological age or the chronological age, then you add this new layer, the age, the face changes a lot with age. So you add this level of precision of phenotyping. So this is just one example, but by combining different data sets, it is well known that you increase the array identification risks. And so with all this cross-border and trans-sectoral sharing of data, I mean, at some point, the accumulation of data is so great that the risk is increasing much. And I'm wondering, actually, I don't want to be alarmist about that, but I'm wondering how we're going to address this issue.
Yeah, and to add to that, I think we need to think about a multi-jurisdictional approach. You've got something happening in one jurisdiction, and then what are the legal nuances or differences across different jurisdictions? That's something else to think about. So I know we've got a question from Chris and another one from Jeff. So Chris, do you want to go next?
Sure. I think I might have jumped the queue. I don't know if Jeff wants to jump in first, but I'll just keep rolling. Just wanting to push you a bit more on some of your comments around informed sort of choice, I guess. On the one hand, you were talking, and this is in response to I guess some of Jeff's comments, but almost like a citizen scientist being able to look into the evidence base and veracity of some of these claims. So assuming, I guess, quite a high level of understanding of these things for the people who are engaging with these direct-to-consumer tests. But then on the other hand, they can't read a 30-minute informed consent form. So I guess, just thinking about that tension. And then whether that approach of the informed consumer choice is already ceding too much ground to the idea that one can have an informed choice about any of these things anyway. So, are we sort of suggesting that you could have an informed choice about something that you actually have no choice or power or control over, and that it is, I guess, just providing a veneer of scientific acceptability to a ruthless commercial enterprise, to maybe put it bluntly?
Yeah, it ties well with Neera's concern, I think, because you've mentioned also, Neera, informed consent, and it is true that we are consenting now for things that we don't really know we don't really know what's going to happen in 5 years from now. Exactly what type of information we're going to be able to detect or acquire using this data. So that's the reason why policies or clear policies regarding the retention of this data, the sharing of this data, and the disposal of biospecimens, why they're important. I think that people can reasonably consent for a limited period of time over certain uses, and that they, if they are being offered the opportunity of being able to retract their data or to delete their data from these databases, I think they should have the choice to do it. I think that it could even lead to people testing more or using more these technologies but it sort of goes in the opposite direction of data sharing and encouraging research and partnership between companies and the public sector. So there's always a tension there, obviously. But I think that since you're using the data of consumers, minimally offering them the choice to either select or agree on secondary uses or agree for a longer period of time retention of this data, I think that's something that it makes more sense to me.
Thanks, Charles. Jeff?
Thanks, Charles. I have I guess a comment and a question. My first is, I guess, back to identifiability. I think often epigenetics is just linked with environment only, but really, as a proportion of variation, genetics accounts for more than shared environment on the epigenome. And so there's going to be some, and I've heard the statistic, 50% of genome-wide association study hits are in regulatory regions, so likely to affect epigenetics. So therefore, epigenetics, and often what we see in our studies, if we see a methylation state at a particular location is influenced by genetics, we see a bimodal distribution. So individuals fall into one group or the other, and it's pretty easy to identify in general which group they fall into, which is basically saying that there's a whole lot of genetic information that we can gather from DNA methylation and other epigenetic data. I haven't actually seen any studies that have quantified that back the other way to actually ask whether those SNPs that link to epigenetic marks can identify individuals are linked with phenotypic traits. So that would be an interesting kind of exercise to go through backwards from epigenetics to genetics to phenotype and see if we can link those. And I guess my second short question is, all those companies you mentioned, are they all multinationals? My suspicion is a lot of them seem to be, but can anybody in any country, I guess depending on law, access these tests?
No. We haven't had the chance to check this for, of course, all the countries, but since we used to travel, I was once in France and tried to access a website of one of these companies, and I couldn't. Well, I could access the website, but in contrast with when I was in Canada, I could not access the page where you would order the test. So there's definitely going to be a difference depending on in which country you are, and yeah, that's something that we would love to look at when we continue our research on that.
And presumably, I mean, in those incidences, they're through VPNs or whatever. I think, as we've seen in China, that if you're restricted, you can still get around those. So I imagine that's not, for those technically savvy, I imagine that's not a barrier.
I heard that in countries that have prescribed direct-to-consumer genetic tests, the people have found ways to get them anyway.
I guess to add to Jeff's comment there, you're thinking about workarounds, there's nothing stopping a citizen of France buying a kit and then handing it over to you, say, Charles, and say, here's the kit. So it would be actually quite easy for that to happen. And I guess it goes back to my concerns around challenges or risks about multi-jurisdictional data breaches of you buying a kit in France, taking it back to Canada, it being used there, and then, well, who owns the data in terms of which country would own that data? And if there was a privacy breach, would it be in Canada, or would it be in France? So it seems that there's, and I could be wrong, perhaps I don't completely understand the nuances of the law here, but it seems that there's quite a vacuum there in terms of something that seems quite innocuous. You buy this, direct-to-consumer kit, and then you use it, and then there's this whole raft of privacy and data breach issues that might occur.
Yes, I mean, it seems to me that it's going to be increasingly difficult to govern data and also to regulate data sharing in a way that can assure people that their privacy is going to be protected. I'm not saying it should not be, but it seems to me that it's going to be increasingly difficult because there are also incentives and good arguments in favor of data sharing. And so I'm wondering, maybe it's a way to work around this. It's to focus a little bit more on making sure that the tests we offer are accurate, that they are precise, that they're validated, and also that down the road, let's say if there has been a privacy breach, we know the best of our institutions have been victims of hackers, and there has been data breaches, privacy breaches, and so maybe it's also a possibility that we should focus maybe a little bit more on protecting people against misuses of the data rather than trying to make sure that privacy is not going to be breached. I mean, I'm not saying that we should not care about privacy, but making sure discrimination doesn't occur is something that can make people maybe more secure about this, or that can I don't know, if you minimize the risks of misuses, then you're less concerned about your information being shared. So at the same time, it's very difficult to discriminate or to say on the basis of what kind of information it should be allowed to discriminate people against,
Yeah, and of course, there's a whole raft of issues that come up with the duty to disclose information. So if you were to, receive particular information about, your genetic makeup or a particular condition, should you have a duty to disclose that information to your loved ones or to your family members? Does it impact them? So there was a case in England, I think it was either last year or this year. It was very, very recent. And it went to the High Court in terms of whether a father had a duty to disclose a genetic condition to his daughter who was pregnant, and would then go on and pass on this condition to her child. So and that's, I guess, somewhat different. I'm digressing slightly, but I guess that raises another whole raft of issues around what are your obligations around protecting your loved ones, or a duty to disclose certain information that arises or may arise from certain tests. So, I mean, I'm opening a can of worms here, but
yeah, we've touched this only briefly when we written with the great team at the International Human Epigenome Consortium. We have a bioethics work group there, and we've published reports to consider on the return of results from epigenetic research, and we started reflecting on, okay, what's different here with epigenetic information, and we were wondering, okay, if people have duty to disclose information to genetic relatives, what would be epigenetic relatives? Yeah. So could there be a duty to disclose epigenetic information to people who live in the same situation as you, who may or maybe have been exposed to similar pollutants, or if you are part of a research project and then you know about this, so is there a duty to say this to possibly also at-risk people? But I mean, you can see that there are so many ethical issues related to these new fields. At first look, sometimes it may seem like it's going to be just again the same thing as in genetics, but it's not that at all.
Sure.
There are similar issues, but there are issues that differ a little bit.
Sure. Another question has just come in, Charles, from Ian Cherist again, and this time Ian is asking, should there be or are there different governance on data sharing in the context of academic research versus commercial contexts?
Well, yeah, it depends, because let's say in Canada, for instance, there's different regulatory regimes. When we talk about what we had in Canada and what we've based our analysis on is a law that's governs data in the context of digital data, and it applies only to commercial entities and to private companies. At the same time, we have ethical guidelines. It's not laws, but we have guidelines for research, of course, and they do not have the same amount of power or the force of law, but they still are followed very closely by researchers in Canada. So it's different regulatory regimes, but they're similar in a way that they aim to protect the privacy of research participants on one hand, and of consumer on the other hand. But
right.
They're not perfect, Sometimes that's the questions we raise, that when guidelines are developed, or that when self-assessment tools, that there's such a document that has been developed by the Office of the Privacy Commissioner to make sure PIPED law in Canada is well applied by direct-to-consumer companies, genetic testing companies. We wanted to look at is this self-assessment tool applying well for companies, not only in genetics, but also in epigenomics and in microbiomics. And so they have nice tools that are not necessarily policies or laws, but that help even not only consumers, but also companies to make sure they comply with the regulations in different countries. So yeah, that's something that companies need to consider, even though they are international in their scope.
Great, thank you. Any other questions, comments?
I had a quick question for, I guess, for both of you. It's about, I guess it ties up a couple of the discussions we've had. Is it possible that you can detect if you detect a social determinant of health in use? For example, your local government or a factory locally has been releasing some toxin that's affected your epigenetics. Can then you invoke, I think it's called tort laws of, can you actually say, well, my epigenetics before the company was there was this, and then after the company moved in, it's this, and this has been shown by researchers to be effective with this. Can then therefore people use this as a way to to sue the companies or the other entities?
Yeah, I think that this is also going to be a field of interest in the coming years. I mean, there's many challenges when you, in tort law in general, when you try to connect the actual source of harm with the actual harm. The causal relationship is often difficult to prove, and the link between the act and the outcome is really difficult to prove. But I mean, depending on the scientific accuracy and the validity of the tests, the precision of the test, it might be of use in court litigations. Yeah, but I don't think it's there yet, at least for most exposures.
Yeah, I agree with Charles. We see it a lot in the law of negligence now in relation to cancer. And you see it in relation to cancer cases that where a person was exposed to asbestos, for instance, from the, say, the 60s or 70s, and now we have the medical science and we have the medical evidence that backs that to say that exposure, 40 years ago or, 50 years ago, 35 years ago, we now have the medical evidence and science to support that exposure to asbestos in your work environment was the cause, or that causal connection, as Charles points out, caused the cancer, and but for your exposure to asbestos, you would not have suffered the cancer. So I think it's a really important point. It's a really interesting point you raised, Jeff, and I think it perhaps may in the future become something where people might bring a cause of action in torts for their environment, that they have been exposed to certain harms.
Can I just on that, I mean, this is something that concerns me about this already being placed within the market and the direct-to-consumer approach, is that the person who, say, discovers this under the scenario Jeff's pointing out, they're a direct-to-consumer fan of these things and they've been using them quite frequently. They notice a change and the website says, well, with this epigenetic change, eat more potassium. So you should eat more bananas to address this issue. And so this individual may go, hey, I'm just going to eat more bananas. Or they may say to their neighbors, hey, we should all eat more bananas. And that will solve the problem. Or if they're even further minded, they might go, hey, we should get rid of this company. But perhaps the company will respond by, we'll just pay for your bananas. And I guess this is concern about putting, I mean, epigenetics does seem to be a powerful tool in some of the social determinants of health, and something we were talking about earlier with, say, intergenerational trauma in relation to colonization and Indigenous justice, is it puts it also in this therapeutic space of turning what is a political and historical justice issue into potentially a therapeutic solution to those problems of whether it's social justice or colonization, I guess is a concern I would have.
Yeah. You mean that I'm not sure about what your concern exactly is, but
well, that it turns what obviously if there's a physical impact on people's health, we want to address the physical impact on people's health. But if that physical impact is also the result of political and social justice, we just don't limit the redress of that to, well, you're healthy now, we've addressed your problem. So to take it to the issue of colonization and Indigenous justice, yes, there are all of these health indices that affect Indigenous communities, but it's not just solving those that are going to redress that historic justice. Or likewise, heavy industry being in poor communities to provide a solution to the diseases as a result of those heavy industries provides one solution, but there are a whole bunch of other issues going on as well.
I think that something that from what I've heard, there is an interest in epigenetics by Indigenous communities in Canada as well. I've been talking with researchers who work with these communities, and I think there's something that definitely has been perceived as a possible way to sort of prove, or as epigenetics could be used as evidence to make people acknowledge that they've suffered trauma in the past, and that the result of this trauma is perpetuating itself, or that it is still with us today. So there's something that is symbolic about epigenetics that I think is worth considering, and this is something we've discussed earlier a little bit. And I mean, we should not understate the fact that epigenetics came after genomics, and that genomics was sort of, even though most scientists will say that they knew for a long time that it was not genetic determinism was wrong, or that geneticization was overstated or overemphasized. People in people's minds, I think that the genetic framing was something that was really a bit fatalistic in a way, and it was knowledge about your biology, but it was saying basically, well, you're like this, and that's the way it is, and good luck with that. So with epigenetics and post-genomic sciences and microbiomics as well, there is something a little bit more. It gives you a little bit more possibility to act upon yourself and your biology, and also not only yourself, but us as a society. When you talk about redressing inequalities, or there's something that's a little bit more hopeful about this, and that it's more enthusiastic. It leads people to be a little bit more enthusiastic, I think, and it's been perceived, at least by some members of the indigenous communities, as something that was better connected with their spiritual and philosophical beliefs. And so maybe, I mean, it's not maybe saying that only epigenetic knowledge from epigenetics is going to be not used or well received, but maybe it sort of balances the fact that we once we're talking only about genetics, and now we're talking also about a biology that's a little bit more flexible, that is reversible in some circumstances, and that we somehow as a society have a little bit more power over, and not only power that is hold or developed by big pharma companies or in the technology sector. It's also, it's also at multiple levels, at the level of society, at the public policy level, that we can sort of see the link between, as you say, the social determinants of health and the biology. So I think there's something. I'm going a little bit in all their directions here, but I think that there's something to be happy about, the fact that we are starting to learn more about these biological processes that are more flexible and more dynamic, and they are being connected with the sciences that have been more interested in cultures, in society, and so I think this leads to interdisciplinary discussions again, and there's hope about this.
Great, thank you, Charles. I think that brings us to the end of our seminar today. Thank you to you, Charles. I know you've given us your evening in Canada there, so thank you very much for a wonderful presentation. It was really, really insightful. It obviously allowed for some really interesting discussion. Thank you to Chris and to Jeff, and thank you to our audience for their questions. And that brings us to the end. So thank you to everybody who attended, and to Charles, Chris, and Jeff. Thank you very much.
Thank you very much.
Thank you.